Hello! Today we'll be learning about the process of transcription and translation. I know, let's start by defining the term transcription. Transcription is the process when information in a DNA sequence, a gene, is copied into a complementary RNA sequence. RNA polymerase is an enzyme from both prokaryotes and eukaryotes that catalyze the synthesis of RNA from a DNA template. There are three steps of transcription: initiation, elongation, and termination. Transcription begins with initiation, which requires a promoter. Promoters tell RNA polymerase 1. Where to start transcription, and 2. Which of the two DNA strands to transcribe and under what conditions. Elongation is the second step in transcription. This is addition of monomers to make a longer RNA or protein during transcription or translation. The third and final step of transcription is termination. This is where the RNA polymerase moves along the DNA until it reaches a termination sequence. At this point, RNA polymerase releases the mRNA polymer and detaches from the DNA. Since proteins are constructed in the cytoplasm, the mRNA must cross the nuclear membrane. Once in the cytoplasm, ribosomes and tRNA, transfer RNA, work together to translate mRNA into a protein. This is done through the process of translation. Before translation can occur, certain modifications must be made to the mRNA. First, introns must be removed through the process of splicing, and second, a 5' cap and a poly A tail must be added to the ends of the mRNA. This clip will help illustrate the process of splicing. tRNA plays a huge role in protein synthesis and translation. It translates the message within the mRNA's nucleotide sequence to a specific amino acid sequence. Joining these sequences together produces a protein. There are three functions of tRNA. There are three sites where tRNA can bind in ribosomes. They are represented in the acronym APE, A-P-E. A stands for the amino acid site, and this is where charged tRNA anticodon binds to the mRNA codon, thus lining up the correct amino acid to be added to the growing polypeptide chain. E stands for the polypeptide site. This is where tRNA adds its amino acid to the polypeptide chain. E stands for exit site. This is where the tRNA, having given up its amino acid, resides before being released from the ribosome and going back to the cytosol to pick up another amino acid to begin the process again. Just like transcription, translation has three steps, starting with initiation. The initiation complex consists of charged tRNA and ribosomal subunits, both bound to mRNA. During elongation, a charged tRNA whose anticodon is complementary to the second codon of the mRNA enters the open A site of the large ribosomal subunit. The subunit is said to have peptidyl transferase activity because it catalyzes two reactions. When a stop codon enters the A site, it binds a protein release factor, which allows hydrolysis of the bond between the peptide chain and the tRNA in the P site. This newly completed polypeptide then separates from the ribosome. In final analysis, the process of protein synthesis usually does not end with translation. Proteins can undergo covalent modifications both during and after translation, with chemical groups being added or parts of the polypeptide chains being removed.
Created using Powtoon.